other end. That's what you're doing. Same way, 3C, you have two ends, you're just using a PCR to know whether my two ends actually are in that space. That's okay. Simple. In 4C, you're fixing one end, and then you're asking what else. So you use one primer for your end, and then you use a random hexamer or something for the other, and then you sequence those to see what you've got. 5C is where you look for multiple genes. So let's say you have 100 genes of interest, and you want to know what my 100 genes in a given pathway or developmental pathway or Wnt pathway or X pathway interact with what all, right? That's 5C. So many to many, okay? And high C is all to all, meaning all chromosome coordinates interacting with all coordinates, right? I explained to you the most complicated process. The other ones are easier to understand, right? You get this or no? So this is how people really look at this. The other methods are called ChiaPET. That's another way that's fairly popular. And then the other method that we use a lot in our lab and other people also do it is called high chip. Basically, where you look for interaction. Let's say you are interested in where the enhancers only interact with the rest of it. So you'll do high C, and then you'll pull it down using an antibody of interest. So I want to know where all enhancers that are marked by H3K27 acetylation interact with. So you will do high C like you will do, okay? And then you'll pull down that thing with a H3K27 acetylation binding peaks, okay? And then you'll see where all that interacted. So we use a lot of that. A lot of people do that because then requires lesser number of reads and very specific question where, because enhancers essentially fold into genes, right? We know that. So that's a most biologically relevant phenomena that one would look at. So you use something called high C and chip together is high chip. Okay, got it? Okay. All right. So let's go back now to this slide. Okay. Okay, so yesterday I told you these loops are maintained by two major proteins. One is CTCF and one is cohesin. We talked about this, right? Should I go back to that, or you guys need a reminder? You're good. So I'll talk in a few slides about CTCF and cohesin because that's really fundamental to the looping architecture and then 3D architecture beyond that. Because once you understand loops, everything else is, just a multiple layers of that, right? To keep it simple for you guys for this time, because when you are more in it, you can look at more other things because it's all confusing and I think it's not entirely figured out. The people are still working on it. Remember, this line of investigation only started around 2010s. So we're very new to this whole 3D genome field, people. So there's a lot of work done in last decade or so, but it takes, a lot of time to figure out a field. Okay. So what is CTCF? I told you CTCF is, what is CTCF? It's a transcription factor. What does transcription factors do? Bind DNA. That's all they know how to do, right? Transcription factors bind DNA. So this is a sequence where CTCF would normally bind. Okay? It's a long sequence. So have you guys looked at things like this? No, probably not, right? So this diagram just tells you, if I were to find 50,000, let's say 100,000 sites in the genome, what percent of those sites would be C, A, T, C, G, C in the first one? So first one is always almost G here, C, A, T, like this. And so you essentially just see what is my probability of finding these sites in that position, okay? That's how you determine what is a binding motif for a given protein. By experiments, you have figured out, okay, that in the experiments where you see probability of finding A or T or G or C at that location. What is the probability? And that is represented in the form of this. So we know this is always C, this is always G, right? Like this, okay? So what did I tell you guys yesterday? That CTCF sites have what do you guys see actually on this? And we talked about this yesterday. Sorry, what? Cytosine methylation. Where is cytosine methylation in this? This is cytosine methylation. So what do you see on this? This site has C's and G's, like highest probability or highest confidence rate. They're always going to be C and G's. So C, G, CpG, when this occurs in this context, there is a methylation. The methylation occurs here or here. Number 2, number 12. Okay, which is the more consensus site. So this is a more of a consensus. Module 2 and 3 that they talk about is a more consensus site. The rest of flanking sites, they can be slightly different. Okay. Did whatever I said, did it make sense to you guys? Yeah? Confusions? Sure? Okay. You guys say yes to everything, but then it will be doubts later. Okay. So this has a certain sequence, right? We got CpGs, methylation happens, CTCF cannot bind. That's been shown very well. On the right side is what? What's there on the right side? Any guesses? Transcription factors bound to DNA. Anybody else? The structural biologist in the group says it is a structure, so we must believe her, right? It is zinc fingers of CTCF. CTCF has 11 zinc fingers. Unfortunately, CTCF structure is not here, which I think I had. Oh, okay. I don't know why CTCF structure is not there. It was here. So CTCF domain structure has around 11 zinc fingers in it. What are zinc fingers? Motifs. What kind of motifs? Huh? What does a motif mean? Okay. That's a DNA binding motif. Zinc fingers bind DNA. Okay. Simple. How many zinc fingers do you see here? 3 to 11. What do you guys see? Can you imagine what is it? What is it trying to show you? It's a very simple thing. What do you guys see on this picture? Is there a DNA there? There is a DNA there, right? And what is not DNA is? That's a zinc finger, and that's part of CTCF. So CTCF zinc finger 3 to 11 bind DNA just like in the major grooves. You see how it's tracking along almost? You see that? How cool is that? There's a protein that essentially it's tracking along major grooves, has built all the zinc fingers in it, right? And can just essentially like go around it and say, okay, I love your DNA, right? That's what it's doing. So this zinc finger bind in a, is this a symmetric sequence, palindromic sequence? Do you guys understand what palindromic means? This is not a palindromic sequence, right? So these zinc fingers have specificity to where they bind, and they have a certain sequence, 3, 4, 5, 6, 7 goes. So basically it only binds the DNA in one direction. That's what I'm trying to tell you. Okay, is that clear? It can only bind DNA in one direction, and that has to 3 will fit here, 4 will fit there, 5 and all. Okay, even though they just bind to the major group. Get it? That's important. That's important. That's the crux of how looping happens in the genome. Okay, what did I say? Okay, before we go there, CTCF binding sites in the genomes are plenty, hundreds and thousands of them. Okay, hundreds of thousands of them. Okay, so this sequence is found fairly commonly. So people have done CTCF ChIP-seq to identify where CTCF binds. It's a fairly abundant protein, so it's easy to ChIP-seq. And they found these low occupied site, medium, and high. And what they find is that the low occupancy sites are always around active genes or in euchromatin regions. So CTCF binds to euchromatin, and high occupancy sites are actually there in the heterochromatin region. So CTCF has all kinds of role, right? It is the organizer of 3D genome. It is the architectural protein that actually makes the 3D genome happen. I made a big statement, but it is one of the major thing. I mean, it's like 70-80% of our 3D genome is because of CTCF. Okay, cool. CTCF is really important. The other molecule that is really important in this space is cohesin. What is cohesin do? Huh? That's right. We talked about this yesterday. We talked about condensins and cohesins, right? What do they do? Condensins have a role. I told you guys this yesterday. What does condensin do? Sorry, what? Okay. What does cohesin do then? What is the difference between condensin and cohesin? Okay. That's right. Right. That's right. Thank you. You guys remember stuff. I see. I like this. You guys get engaged, which is great. Cohesin has a structure which is shown on your screen. I'm going to try to explain that a little bit more because cohesin is the other really key protein along with CTCF. Only CTCF cohesin work together to make the looping. Condensin is actually not very involved in this process for whatever reason. I don't think people understand that. So some people have shown that condensin also is involved in 3D chromatin structure, but cohesin is a primary driver along with. And that's my objective to tell you guys what they do in next 5, 10 minutes, 15 minutes before we end this session. So cohesin has this structure where you see on the top there is a hinge domain, SMC1A, SMC3. These are coil proteins. Okay, essentially they're flexible coil coils. They're like your spring. Okay, two springs like this, my two hands like this. Okay, there's a hinge on top. What does hinge mean? It's a hinge, and on the bottom there is ATPase heads. There are two proteins that they're ATPase heads. There's a third unit called RAD21, and there is STAG1, STAG2. The shapes of them actually tell you the shapes of those proteins. They are like that. Okay, so the shapes are critical on this particular protein because essentially cohesin is a ring like this. It's a ring like this. It has to open up, close, open up, close. Okay, it can be broken down as he said earlier in metaphase for the chromosomes to separate in anaphase. Okay, cool. Yeah, PDS5A and 5B are two proteins that are involved typically in the cohesion process during the cell cycle. And then they can be replaced. So these arrows mean they can be replaced by this NIPBL and MAUT2 protein. Okay. Good. NIPBL is binding to this in the 3D chromatin space, which is what we are interested in here. It's absolutely crucial for ATP hydrolysis to happen. It also helps in this thing bind DNA. Okay. All right. That will become important in a second. But did you guys get this? It's a beautiful little protein. Guy called, as I said, Kim Nesmith in Austria did a lot of work on these, really figured out the structure. Obviously, a lot of people have done that, just one of the famous guys. This is other proteins. So there is cohesin, similar to that is condensin, similar to that is SMC56. Okay, there are two condensins, 1 and 2. They all have similar structure. I want you to appreciate they're all ring-like structure. They're essentially like a ring clasp. Okay, they'll do what they have to do. Okay. I want to play this video for you guys. This is a paper. I find the paper very interesting. You'll appreciate this. Of course, if I can play it here. Why is the sound? Huh? Oh, is the light off? The power off? Cool. Cell Reports is a very interesting, important journal. You know what? Why? Because it's open access to everybody. You don't have to look for subscription. I published several papers in Okay, I want to show you guys these videos. Okay. You guys see this? This is your DNA as is by something called atomic force microscopy, AFM. It's a very interesting technology. I don't know the details of it. That's so I'm not going to even try to explain what this means, but you can read if you want. And then you look at this video. What do you see as difference? Say that again. I can't hear. You guys have to speak up. There are no wrong answers, remember? Let's play this one first. This is literally DNA with treated with topoisomerase, because topoisomerase will coil it more, but so topoisomerase will uncoil it a little bit. But you see that? And I'm going to show you the second video. Watch carefully. Great. What else do you guys see? Great. Do you guys agree there is more loops in this? Is there more structured to this? Okay. This is essentially atomic force microscopy in the absence or presence of cohesin. How cool is that? You put cohesin in, it makes coils like that, right? That's what I want to show you. This is an experimental evidence to say that cohesin does what it does, right? Cool? Do you guys find this cool? The fact that you can even just image the DNA like that is really cool. Okay. Okay, this is from the same paper. Essentially, they're trying to look at the mechanism of how cohesin will make loops. Okay. So you agree that cohesin makes loop, minus plus cohesin, it makes loops. This arrow tells you that it's gonna twist the DNA. And what happens when you twist the DNA? How do you put something in a sui dhaga? Or how do you make a loop from a thread? You have to hold one end, you have to move the other hand so that it can make a loop, right? That's exactly what cohesin does. Okay? Beautiful little machine, right? Functions all the time. That hinge is holding one end. There is ATPase here. Remember, ATPase is on this end. ATP function allows the opening of the thing. Okay, we'll come to that. Let me show you this. This is how things work. I'll come to that model. But essentially, cohesin can be loaded on DNA. There are two CTFs of size. One CTCF binding site is CBS, essentially. Okay, CBS is CTCF binding site in forward direction, in reverse direction. Okay, so forward direction, reverse direction. Okay. 2, a loop passes through the cohesin by extrusion. So cohesin will bind to any DNA. It's not specific to anything. It binds all DNA. Okay. That is needed, of course, right? Cohesin has to just do this all the time. So cohesin then will bind one side, make a loop like this, right? Keep doing that till it hits the CTCFs, 2 CTCF sites, which are in inverse direction, not in the same direction. A same direction, CTCF cannot bind to each other. CTCF this way will actually bind and stop, because 2 molecules of CTCF are now come together and will take this to bind, and ring-like structure will essentially hold it, clasp it, that's it. That's your loop. Not clasp it, but that's your loop. You get it? So basically, it's a combined action of two complexes, CTCF protein and a cohesin complex, that works together to make loops. This is just the mechanism of 3D looping. Very nice, right? They said beautiful little machines our cells are made, and the code they made. So the two CTCF sites, if they're in the same direction, are not going to make a loop. The CTCF sites in the inverted direction only is going to make a loop. Okay, that's it. So that's how a lot of this happens. So it's going to keep going. Some of these bindings cannot be, they are not that strong in real world. So some of this will go back, and there'll be more looping occur at a later position. So that's why the multiple loops become. So it's the intensity of the interaction between the two CTCF sites and surrounding chromatin around it that will ensure whether this binding of inverted repeat loops are actually same or not. Will stay, and how long will that stay, how much will that be stabilized or not, and that depends on chromatin marking and all of that. Okay, so not going into that, because again that's complicated, there's a lot more to it. K27 trimethyl plays an important role. I told you guys earlier, ubiquitination plays an important role in all of that processes. Histone H1 plays an important role. Okay, so this is the same thing shown another way, and I guess you would appreciate the same thing, right? Okay, more simplistic possibly. I mean, they're from two different review articles showing the same thing. I didn't know which one you guys will appreciate a little more, right? I just wanted to put it out there. Same thing, CTCF cohesin extrusion. This is called loop extrusion through the cohesin ring. Okay, and this is going a little bit more into the mechanism. As I told you earlier, this is the heads hinge. Heads are engaged, okay, together. ATP hydrolysis happens, the heads get disengaged, the thing opens, okay. So the whole thing actually opens and closes based on ATP hydrolysis. So every single, yes, ma'am. What becomes outside the loop? And from that loop, in the both side, it is showing the cohesion. So how they are coming back to their position? How they are coming back to their position? These things. Yes. It's a simple breakage of cohesin ring, right? So the other proteins I was showing you around cohesin actually will cause the breakage of cohesin at some point. So that's where the modifications are important, and regulation of this whole process is important. So let me go back and see. These proteins, right, when this NIBL is there, it will essentially help the ATP hydrolysis. Without that, it cannot reproduce. So NIBPL is the regulatory protein of this that will essentially has to bind to do that. Okay. Then whenever there is another protein called VAPL, this protein, when they binds, it actually breaks. That's actually a regulator of breakage and all that. If I remember correctly, there is more to it than I know. Okay. I mean, I don't remember all things that people have shown now, but you can look up. My knowledge is not like 100% up to date, guys, right? As you can tell, I have too much in my brain and I can only hold so much. I'm a little human, but you can always go and read. My job here is to tell you the basics of things, and then you get the details yourself. Okay? I'm not at all pretending that I know everything. I don't. Okay? I also sometimes read and tell you guys what I'm actually, because it's just difficult to keep track of everything in the literature. And these things keep changing because it's very, it's a field which is every day people are working. There are new things, new hypotheses. But it's a very cool field. I mean, I just loved it when I saw it. I started working on this right at 2011 when I saw the first paper. And it was like, I want to solve this in cancer. We've been at it for 10 years. We're going to publish our first paper now after 15 years. It's been a challenging field, but it's a very, very, very interesting field. Yeah? This is one of the ways how cancer translocations happen. because things that are too far apart, they actually will be together because of 3D chromatin structure. Then some DNA repair damage process will happen, and those will translocate. That's how translocations happen. And in fact, this high C method is used now to find translocations, because any off-diagonal big red signal actually means translocation. It's not a 3D interaction. 3D interactions are not that strong. They're strong, but they're not like, so on the high C map, go back to this for a second. Here, if you see anything which is like this here, you see this? These are translocations. This whole chromosome, this region is actually translocated to this. It's actually not a 3D interaction. It's not a normal interaction. They've actually joined together, actually physically. Okay. So anything that shows as red as off diagonal a lot of times is actually a translocation. People are using this a lot in cancer world to identify new translocations, because otherwise it's very difficult to identify those translocations. You get the translocation piece, right, where one DNA is actually joined to the other DNA. And you can tell, obviously, the red and the blue, if they were joined, they'll be together, right? So off diagonals are, sorry, I think I switched in between, but that's an important point. Okay, cool. Let's go back to this. So basically, ATP hydrolysis is important for these two balls to come together or not, right? And that's how they stretch or not. Very simple. Not very simple, it's more complicated. This is the model in which it apparently works, where you see this, you see the loops right here. The blue, yellow is essentially the DNA. Okay, there is two heads. One hinge is binding here. This is binding here. Okay, so one, and this place is okay. So the DNA gripping, so they're binding the DNA in two ends. DNA slipping, the DNA slips through if the binding is not strong enough here. Okay, and the Brownian ratchet, when you do the ATP hydrolysis, the one of them opens, and then there's a Brownian motion. DNA just moves, just like your thread, right? If you hold one end and you just, let's say you're holding two like this, and there's a spring, because it's a coiled, right? Everything is coiled, right? And there's a, I mean, DNA cannot be just held like this. It requires proper energy to be held, because it has, what is it called? Because it's a coiled structure, it has a, what is it called in physics? There is energy, right? I mean, otherwise it will open up, just like your spring. Spring cannot just be held like this, right? Same thing with DNA. So once you once you not bind one side, because you're essentially binding DNA at both sides. If you open one, then the DNA will just go, right? And then, so this is what the hydrolysis does. Hydrolysis opens it up, the DNA goes, and then it binds again, because the ATP molecule is only so much, right? And that they come together because of these other proteins that I showed you, RAD21 and NIBL, and also NIBPL has to bind here. This hydrolysis has to occur. Then the DNA binds again, releases, then grips again. So you found the loop, essentially. Okay, that looping is random until it hits the CTCF sites. So this extrusion will keep happening till it hits the CTCF sites, where the ring cannot do anymore anything. Okay, and that is shown in this slide, I think. Yeah. Oh, my picture is not here. That's why, because some of the pictures that I post here are not here. Oh, there is a whole picture here. I don't know why my slides, when I send to, because I work on a Mac, I think when I send this, some of the images don't show up. Yeah, you can see that, right? My image is not there. So here, I mean, this is more complicated stuff, and I think you don't need to worry about it. But I could show it next lecture. I can show this. But basically, there is models on how and why when CTCF, when it hits the CTCF. Now the question is, okay, when it hits the CTCF site, when coenzyme and CTCF hit each other, how does this block, right? Like, why does it block to begin with? And how do you make multiple loops? And how do you make the TAD structure? So people are still figuring that out. It's not well known yet. But basically, there are some models on how CTCF binding itself to the chromatin structure will displace some of the proteins, and the cohesin binding will become stronger. It won't be able to go, NIPBL binding will not be allowed anymore. So ATP hydrolysis won't happen. So that just holds on there, something like that. Okay, but we can talk about this more later. Okay, all right. I think we'll end there for now, but we'll come back around 12:10 or so, 5:10. Okay, and we'll go to the next round. Okay, cool. I just want to keep it simple for you guys, because I also didn't make all the slides completely. Good. But I hope this sort of illuminated you guys a little bit more. We can come back to, yeah, I mean this, oh, actually, sorry, last slide here. So I put this table here. When you guys get the slide, you will have it. But this is essentially a summary table, which one of my postdocs had made earlier, that all the tools, computational tools, or the methods that are used for probing these, and there are lots of tables like this, and other people can find what methods help us probe what part of the epigenome, and what computational tools you will use to actually do the analysis. If that is useful to you guys, you can look at it. And so there are multiple tools out there. And I mentioned this method called high chip, which you see on the bottom, right? That's one. So there are multiple methods, and you essentially have people have, computational biology, this is what they do, right? They make new methods to analyze the data, and that's that. So you can get that, and that it's all omictools.com and whatever, right? So you can check it out. Okay? All right, thank you. I'll see you in about 15, 20 minutes, okay? Maybe let's say 12:10. Okay? Let me see what you guys write. Thank you. Yes. Oh, I have to make this slide for the next one.